Hello, my name is Tyson Greener. The date is July 15th of 2022. I chose two different case studies, and I'm actually going to do case study number 3 and then case study number 1, do it a little bit backwards just because of the way I have it typed out here. The first case study that I'm going to go over, case study 3, talks about a 47-year-old man presents with severe pain to his right great toe. He is unable to bear weight on his right foot, and there was no apparent trauma. He reports pain started last night and continued to increase with severity. Based on his physical exam and laboratory findings, he is diagnosed with gout. Gout can be caused by several different mechanisms, and blood samples are drawn and urinalysis is performed. The results from this individual are listed below. And it tells me my plasma urate normal levels are less than 7, his are 10.5, and then urinary uric acid normal levels are 413 plus or minus 78, and his are 215. Then goes over and says, what is the best explanation for the presentation of gout in this individual, and please explain why the other choices are not consistent with the data or what additional information you may need to decide the root cause. And then it asks overproduction of purines, decreased salvage of purines, or decreased urinary excretion of uric acid. So I'm going to go through each one of those three first and just ask maybe some other information I would like to know before I decide which one, and then tell you which one I think it is based on the information that I have and the research that I have done. So overproduction of purines, obviously we know that can be caused by many things. Purine's base is comes from the pentose phosphate pathway, which is your ribose 5 sugar. And usually the overproduction of purines comes from, or a lot of times it can come from, the increase of that sugar being overproduced, or its other pathway being shunted or blocked. So I would like to know maybe some other blood labs of this patient, as well as what does the patient's diet look like. You know, if he's overproducing all the sugar, or of this ribose 5 sugar, is his diet recently changed? Is he eating a lot more sugar than he normally is? Things like that. I'd also like to know if there's a deficiency in any other enzymes, such as talked about in the videos, the glucose 6-phosphatase deficiency can also show, have an increase in the production of those ribose 5 sugars that are overproduced, which can cause that overproduction of purines. There's then the decreased salvage of purines. This wasn't talked a lot about a lot, and the research I did, I couldn't find a whole lot. But what I did find was that usually, if this is the case, the patient presents with lots of other symptoms. From the case we have here, it doesn't seem like he's presenting with any other symptoms except for pain in the right great toe, and unable to bear weight on that foot. There doesn't seem to be a lot of other neurological or cardiac or any other signs and symptoms that we may be looking for if there was a decreased salvage of purines, which then leads to a decreased purine usage, basically meaning that there's things that your DNA or transcription means that you need that aren't happening because of your decreased level of purines within the body. The third one, decreased urinary excretion of uric acid. The big thing I'd like to know, does he consume alcohol? How much alcohol does he consume? Things like that, and what's causing those things. So the best explanation, in my opinion, from the information that we have here of an elevated plasma urate level and then a decreased urinary uric acid level, to me almost seems like a combination of decreased urinary excretion of uric acid as well as an overproduction of purines. We find later in the case study that the patient recently switched from diet soda to regular soda, and he drinks 4 to 5 liters a day. That tells me, with that information, that this is an overproduction of purine issue. which I kind of talked about a little bit earlier, but you have that increase in sugar intake, which is a ton of sugar. All of a sudden, your ribose 5 sugar is going to increase significantly, and you're going to have an overproduction of that, which is going to lead to a buildup of your uric acid in your body, which is probably what's causing his gout in this situation. So that is it for case study 3, I believe. I'm going to just make sure that's correct. Perfect. So on to case 1. Case 1 is talks about a newborn girl appeared at birth, but within 24 hours she developed lethargy, hypothermia, and apnea. The attending physician suspects a metabolic disorder and orders a panel of labs. A full blood workup was done for this female, and the data is presented below. The initial ammonia level was 314 and rapidly rose to 600. And so we're going to discuss kind of the mechanisms of ammonia transfer in the body that should be occurring, talking about the different enzymes that are used. So basically, ammonia transfer is tricky within the body because ammonia is very toxic to the body, so it has to be transferred, and it's usually done so through alanine and glutamine, which are non-toxic in the bloodstream. But you have to get those ammonia particles within the body, which are produced from amino acid breakdown, because that's what the body uses for fuel. You have to take that ammonia out of the body, out of the tissues, through the bloodstream into the liver, where it can then be broken down and excreted through the urea cycle, and then excreted through uric acid through the urine. So in order to do that, there are 3 main enzymes, as you will, that help transfer this and then break it down. The first one being glutamate dehydrogenase, present in most tissues. The interesting thing is it can fix and free ammonia, so it helps in both areas depending on where we need the ammonia. So it will fix the ammonia using NADPH from alpha-ketoglutarate and create glutamate using that NADPH. This glutamate is then used by glutamate synthetase, where it fixes another ammonia group out of the glutamate, forming your glutamine, which is then safe to travel through the bloodstream, carrying those 2 ammonia groups. This is a process that requires ATP, which is interesting because this process is not regulated by hormones, so it's active in both the fed and the fasted state. So the ATP that is used for that, it's either coming through the citric acid cycle in the fed state or beta oxidation in the fasted state. So once it fixes those 2 ammonia groups, onto ATP, from ATP, onto glutamine, it is then carried through the bloodstream back into the liver, where glutaminase becomes involved, which is our 3rd enzyme, and it is active in the liver. It is responsible for the deamination of glutamine that was synthesized by glutamine synthetase. That then it frees the first ammonia, if that makes sense. So it goes back from glutamate to glutamine. From glutamine to glutamate, thank you, in the liver, which then frees that first ammonia, which then enters the urea cycle to be excreted in the liver. Then, I lost my spot. The glutamate then enters a reaction with glutamate dehydrogenase again, which, as we talked about before, can fix and free ammonia. Then at this point, frees the second ammonia in the liver, and that's when you have that second ammonia freed, which also then goes to the urea cycle, and then that glutamate goes back to becoming alpha-ketoglutarate. This alpha-ketoglutarate is important to note, just for a quick minute, that alpha-ketoglutarate can then become pyruvate, which then can be used in many other places in the body, whether that's gluconeogenesis or the citric acid cycle, to become acetyl-CoA. I'll all these different areas that this enzyme can be used. So that's something to keep in mind on. Before we move on, we need to discuss the role of transamination. I'm running out of time, so I'll be quick. But transamination is the process where the nitrogen is transferred to an amino acid to be shuttled through the body. So transamination takes place through those three different enzymes that we talked about: glutamate dehydrogenase, glutamine synthetase, and glutaminase. The biggest ones we look at that are clinically relevant are AST and ALT within the liver, because if those are elevated, it shows us that usually that there is an issue within the liver and with the transamination process. Alanine is transaminated pyruvate through the use of alpha-ketoglutarate, which I talked about earlier. Anyway, the next question is, how is nitrogen transport connected to gluconeogenesis in the fasted state? I touched on this briefly earlier. Nitrogen helps to create the intermediate amino acid that are used for gluconeogenesis. So through nitrogen transfer, we produce alpha-ketoglutarate, which can then produce pyruvate, which we know pyruvate is essential in our process of gluconeogenesis. States then that further analysis shows that the urine uric acid levels was markedly elevated, and the plasma sertraline levels was normal or low. And then it asks, in this individual where uric acid levels are elevated, what urea cycle enzyme that is most likely deficient and what urea cycle enzyme is least likely to be impaired? Justify your answers. So the urea cycle enzyme that is most likely deficient is OCT, and it can be seen because of the elevation of the uric acid. Normal to low citrulline levels and an elevated ammonia level also kind of supports that the OTC enzyme is the one that is impaired. I think through my studies that the amino acid then becomes essential is sertraline, because if that enzyme OTC is then what's low, your sertraline is then going to become as low as well, which is then going to be your essential amino acid in this case. That's it. Thank you for listening, and until next time.